Hello everyone. So we are going to discuss about glycogenesis. Okay. Glucose and here is glycogen. So this is reversible. Glucose to glycogen. This is glycogenesis. This occurs during energy-rich state. And glycogenolysis. This is production. This is catabolism, formation of glucose from glycogen. This occurs during fasting. And let us know the definition. Glycogenesis is the formation of glycogen from glucose during energy rich state. Okay. And sites of this reaction. It occurs in tissue sites. In tissue sites, it occurs in liver and muscle. And intracellular environment, it occurs in cytoplasm. Okay. Then this is reaction pathway. Number A, activation of glucose. In activation of glucose, glucose is activated, that is UDP glucose is formed. Okay. First, this glucose is converted to glucose 6-phosphate. Sorry for this overwriting. Okay. This is up to this it is similar to glycolysis. Okay. This reaction is catalyzed by these two enzymes, hexokinase and glucokinase. Hexokinase is more strong and glucokinase is less strong. Okay. Both of these actually form the same thing, but the conditions in which hexokinase act favor glycolysis, and the conditions in which glucokinase acts, it favor the glycogenesis, because glucokinase has higher Michaelis constant, because it has less affinity, it is less stronger. It is not found everywhere, but this is found everywhere. This is found mostly in liver, where liver generally stores this excess glucose. This glucokinase is generally used in glycogenesis. And from glucose 6-phosphate, there are several ways. One way is towards glycolysis, and this way is towards glycogenesis. In this reaction, glucose 6-phosphate forms glucose 1-phosphate, and this is reversible, conducted by this enzyme phosphoglucomutase. Here the phosphate group has changed the position from 6 carbon to 1 carbon. So 1 carbon. I have tried to show the pathway by which this reaction occurs. Here, this the lone pair. It has a lone pair. Okay. There is a nucleophilic attack on this phosphorus atom, and there is resonance. Then this bond breaks, and the pyrophosphate this detaches. This is shortened as PPI. This reaction pathway is actually not important from exam point of view, but knowing this makes biochemistry interesting. And here we get the activated glucose, which is UDP glucose and pyrophosphate. Then we have lengthening of chain. Two enzymes are important here. The two enzymes are glycogen synthase, and there is a branching enzyme which is amylo 1,4,2,1,6 transglucosidase. Glycogen synthase, it This is responsible for a straight chain. It requires a glycogen primer, which is called glycogenin, and the UDP from the activated glucose is removed here, and glycogen amylase actually continues to form. Continues to elongate. Then there is branching enzyme, which is responsible for branching when the chain length is increased by minimum of 11 glucose residues. A part of the alpha 1,4 chain, minimum of 6 glucose residues, is transferred to neighbor chain, producing the branching 1,6 alpha 1,6 linkage, and these branches which are produced grow further by the glycogen synthase in different directions. Now, significance. It is a storage polysaccharide and stored in liver and muscles. Regulation of glycogen synthesis. Glycogen synthase is the regulatory enzyme. It is regulated by covalent modification, which is more precisely reversible covalent modification of glycogen synthase. This means reversible covalent modification means addition or removal of same type of group, commonly phosphorylated form. And here, glycogen synthase, it is active when dephosphorylated. It phosphorylates by any kinase, any protein kinase, and it becomes inactive, and phosphatase removes the phosphate and it is activated. Okay, that's all for this part. Sorry for any disturbance, and we can take a glance